We see the headlines literally every single day, right? Artificial intelligence is going to cure cancer, or machine learning discovers a massive new biomarker. But how does that actually happen? How do you take a massive, buzzword-heavy concept like AI and turn it into a targeted, scientifically sound research study that can actually survive peer review? Spoiler alert, it doesn't happen by magic. It happens through meticulous, rigorous design.
So, welcome to this explainer. Today we are doing something pretty unique and honestly super exciting. We're going to dissect an actual real-world feedback letter from a professor to a computational biology student. We get to look right over the mentor's shoulder and see exactly how they tear down a vague initial idea and rebuild it into a rock-solid research proposal. If you've ever wanted to know how high-level medical AI research is structured before a single line of code is even written, you are absolutely in the right place. Let's dive in.
Here is our blueprint for today. We'll start with refining the research focus, then identifying the research gap, formulating a strategic dataset plan, mapping the ML workflow, setting scientific boundaries, and finally defining those crucial next steps.
Okay, Part 1. Refining the focus by totally stripping away the fluff.
The absolute first step of any serious scientific endeavor is stripping away those broad concepts that sound amazing in a pitch, but mean absolutely nothing in a lab. Just look at the sheer contrast here. The student's original idea was simply AI-driven biomarker discovery. The professor's response? Yeah, way too vague. Instead, check out the laser-like precision of the approved title, Explainable Machine Learning-Based Prioritization of Transcriptomic Biomarkers for Early-Stage Cervical Squamous Cell Carcinoma using GEO and TCGA-CESC datasets. Phew, that is a mouthful, but it tells you exactly the how, what, when, and where. This is what rigorous science actually looks like.
Moving right along to Part 2. Identifying the research gap, or basically, finding your niche.
See, once you have a precise title, you actually need a reason to exist. The mentor points out a pretty critical reality here. The field is crowded. A bunch of studies have already used these exact datasets to look for cervical cancer biomarkers. But the professor smartly guides the student away from saturated areas like general diagnosis, immune signatures, or treatment response. So the real scientific gap, the untapped potential, lies in focusing specifically on early-stage or precancerous cervical lesions, and pairing that with explainable machine learning. We aren't trying to diagnose cancer that's already glaringly obvious. We're trying to prioritize candidate early biomarkers. Huge difference.
All right, Part 3, a strategic dataset plan. These are your raw materials.
In computational biology, your raw materials are your datasets, and this mentor is incredibly strict about how they're used, which brilliantly shows us how a data hierarchy works. You can't just toss all your data into one big pot and stir. The professor organizes these datasets with surgical precision. For example, GSE 63514 is your main discovery dataset. It has normal, CIN, and cancer samples. Once you find a signal there, you test it in GSE 7803 for external validation, just to be absolutely sure it wasn't a fluke. GSE 64217, that one is relegated to just supportive validation because its sample size is simply too small to be statistically powerful on its own. And finally, TCGA-CESC is used for clinical relevance and stage comparison. Every single piece of data has a highly specific, defined job to do.
But wait, here is a massive red flag the mentor raises. And honestly, ignoring this could ruin the entire study. The professor explicitly warns the student, do not use the TCGA-CESC dataset alone for early detection. Why? Well, because that dataset mainly contains invasive cancer cases. Think about it. If you're trying to find early precancerous biomarkers, but you train your model on invasive cancer data, your results are going to be completely skewed. It's a fantastic reality check. You have to know the actual biological reality of the data you're computing.
Which brings us to Part 4, the machine learning workflow, where we actually extract meaning.
With the data organized safely, we can map out the core machine learning workflow. The professor lays out an expected methodology here, and it reads like a perfect 6-step funnel. First, you download and pre-process those GEO transcriptomic datasets. Second, you identify the differentially expressed genes, literally finding the genetic differences between normal, precancerous, and cancer groups. Third, you bring in the heavy hitters, applying machine learning models like random forest, SVM, and XGBoost to select candidate genes. Fourth, you explain the AI using SHAP or LIME. Fifth, you validate your top genes in that independent dataset we just talked about. And finally, step 6, you step away from the keyboard and check the biological relevance using pathway enrichment and literature support. because at the end of the day, it actually has to make biological sense.
I want to pause on step 4 for a second, because it is absolutely vital. The professor specifically demands explainable AI. In medicine, an AI cannot be a black box. You can't just say, well, the computer told me these genes are important. No way. You have to know why. Using techniques like SHAP or LIME is a total non-negotiable step because they pry open that black box and tell the researcher exactly which features drove the algorithm's choices. That is exactly how you build trust in the science.
Let's look at part 5, setting scientific boundaries. Time for a reality check. This next section is arguably the most critical part of the entire feedback letter. The professor gives a very explicit do not claim list. This study cannot claim clinical validation. It cannot claim it's developing a new diagnostic test. It cannot claim patient-level clinical application, or confirmed biomarker discovery, or experimental validation, unless actual physical lab work is done. It is so crucial to grasp the scientific difference between a computer-simulated hypothesis and a real-world clinical reality. Claiming your algorithm is a clinical test before it ever hits a wet lab? Yeah, that's a massive scientific faux pas.
To make absolutely sure there's no misunderstanding, the mentor literally spoon-feeds the student the exact wording they have to use. You must present the final genes only as candidates. You have to call it an in-silico prioritization of candidate transcriptomic biomarkers, or a hypothesis-generating computational biomarker study. By the way, in silico just means performed on a computer or via computer simulation. The professor is strictly enforcing intellectual humility here. The computer points the way, but the lab confirms the destination.
And finally, part 6, defining the next steps and the green light requirements. Okay, so the idea is refined, the data is planned out, and the guardrails are firmly in place. But to actually get the green light to start, the professor demands a 1 to 2 page concept note. This isn't just administrative busywork, it's a contract of methodology. Before final approval, the student has to put down on paper their final title, aims, selected datasets with their accession numbers, planned software expected outcomes, and most importantly, the limitations of their study. They essentially have to prove they understand the assignment. And right smack in the middle of that concept note requirement is this brilliant point of scientific rigor. The student must provide the exact definition of the word early. Now, in casual conversation, we all know what early means, right? But in research, early means absolutely nothing until it is strictly defined by the sample groups. Are you comparing precancerous CIN or HSIL lesions against normal tissue? Or are you comparing stage 1 early cancer against stage 4 advanced cancer? The computational model will only do exactly what you tell it to do, so your definitions have to be absolutely flawless.
And that brings us to the end of the professor's blueprint. Honestly, it's fascinating to see just how much rigorous, disciplined thought goes into a study before a single algorithm is even run. But it leaves us with an incredibly exciting thought. If this strict, meticulous in silico framework is just the hypothesis-generating phase, what happens next? What does the future of medicine actually look like when these highly vetted, AI-prioritized candidate biomarkers make it to the wet lab and eventually to the clinic? Because that rigorous computer simulation, that is the vital first spark for modern medical breakthroughs. Thanks for joining me on this explainer, and keep questioning the science behind the headlines.